welcome back. Today we're going to talk about transformation and oncogenesis, or how viruses cause cancer. These two words are very different, so we need to explore those first. So let's do an experiment. On the left, we take a chicken embryo, a hamster embryo, doesn't really matter. You remove the head and the legs, and then what's left you chop up and you treat it with trypsin to make single cells, which you then plate on a plastic dish, and the cells adhere. And on the dish on the left, we just leave them untreated, and the cells will make what we call a monolayer. They will sit down on the plastic and behave very nicely. When they touch each other, they will stop growing, and eventually they fill in all the space on the monolayer, on the plastic, and stop growing. But on the right, we have mutagenized the cell. You can do that with a chemical mutagen. You can do it with ultraviolet light. And then every now and then on that nice monolayer of cells, you will see cells that pile up. And if you look at them under the microscope on the right, you can see they behave very differently. We call these transformed cells. And that is what we're going to explore today. This is transformed by mutagenesis, but we can also transform cells using viruses. And we have to visit the ends of chromosomes and the enzyme that maintains them. Now, if you remember when we talked about DNA replication, if you have a linear double-stranded DNA, you have an end problem, because if you remove the RNA primers at the ends, eventually the ends are going to get shorter and shorter and shorter. And the solution to that in viruses, we talked about having circular genomes, for example, having self-priming. But in our genome, the solution is telomeres. These are repeated sequences at the ends of chromosomes that are put there as you're developing as an embryo by reverse transcriptase. And so there on the right, you can see the end of a chromosome. It gets degraded as you replicate, because it's single-stranded, and the host doesn't like to have single strands around. So the solution is a telomere, a number of proteins, including telomerase, which is a reverse transcriptase, actually, takes a small piece of RNA and copies it over and over and over again to make long ends on your chromosomes. And when you are born, telomerase is inactive. It's no longer active, and as you age, your chromosomes get shorter and shorter and shorter, and that's part of senescence. Getting old is losing the ends of your chromosomes. All right, now, in culture, you can put cells in culture, as we just did. Every time they divide, which is about 24 hours, the ends of the chromosomes get a little bit shorter. And after you lose about 4,000 base pairs, the cells die. And so human cells will only last about 50 divisions because of that. Mouse cells will last a bit longer because they have longer telomeres. And that's going to be important in a moment. Every time DNA replicates, every time a cell divides, it sustains mutations, because DNA replication is error-prone, not as much as RNA replication, but it is error-prone. And so the longer you let a cell divide, the more the chance is it will become immortal. So we'll explore this in a moment. So here's another way to look at this. On the left, there's your chromosome. The ends are in blue. Those are the telomeres. As you age, you have telomere shortening. And at a certain point, the cell undergoes apoptosis when the ends get too short. And that's senescence, that's aging, that's part of aging. On the right is a cancer cell. Cancer cell, in this case, has an active telomerase. It hasn't been shut off. It may have been turned back on again by mutations, sustained by cigarette smoking, UV light, whatever. And their cells divide forever. And when cells divide forever, that is a recipe for transformation and oncogenesis, which is the formation of a tumor. So this is behind what we're going to talk about today. So here's the establishment of a cell in culture. We've taken our cells. I showed you hamster cells, but it could very well be mouse cells. You could do this with human cells. You just take a little Q-tip and swab the inside of your cheek. You get a lot of cells off of it. You can put them in cells in culture, and they will grow. On the top is your mouse cells. So every day you split, the cells divide, and you keep the cells going. They will slow down in their growth as they age. They're going through senescence, but they don't die. They go through a crisis period, and then some mutant cells arise that are now immortal. So because the mouse cells have been able to divide for so long, they've sustained enough mutations to make them immortal. And one such mutation could be to reactivate telomerase, but it could also be in many other things as well. Now, human cells don't do that. They grow for a while and they die because the ends of the chromosome are shorter than mouse cell ends, and so they don't have enough time to sustain the mutations. If you want to make immortal human cells, you have to do something to them. You could put a gene for telomerase in them, and as you're going to see today, you could mutagenize them. And as we'll see today, you could also put the gene for T antigen in them, and that will immortalize them. And you may be thinking, what? T antigen is involved in origins and transcription, but it's also involved in this. Transformed cells, they grow indefinitely like HeLa cells. They were taken from a lady in the 50s who had a tumor on her cervix. They're put in culture. They were the first cell to grow forever. You know, this was an amazing discovery, and HeLa cells were used for years. So they grow indefinitely. They lose their anchorage dependence. So cells, in order to grow, have to stick to the plastic dish. They don't like floating. If you put them in agar, for example, where they're floating, they don't like it normally. But if they're transformed, they will grow. They lose contact inhibition. So as I said, the untransformed cells, when they touch each other, they stop growing. But if they're transformed, they keep growing, and you see they pile up on top of one another in that side view in panel B. They'll form colonies in semi-solid media. That's a reiteration of anchorage dependence. And they have decreased requirements for growth factors. If any of you have done cell culture in the lab, you know you have to make up a medium, which is typically red, and you add stuff to it, including serum, which you can buy from. You can buy bovine serum and all kinds of serum, right? So after they use the cows for other things, they take the serum and they sell it. It's full of growth factors, and it makes the cells divide. And transformed cells have reduced requirement for growth factors. So that's transformation, a change in the fundamental properties of the cell that I just described. But oncogenesis is different. Oncogenesis is the development of cancer. And a tumor, of course, is a swelling in you caused by abnormal growth of cells. A tumor can be benign, in which case it won't kill you, or it could be malignant. It can metastasize throughout your body. Cancer is a genetic disease, 8 million deaths a year in developed countries. Why don't we look elsewhere? They don't have good enough health care to know, but there certainly is cancers in other countries as well. And cancer arises when you have mutations, about 12 or so, in genes that govern cell division, including telomerase. You can have a mutation in the telomerase, but we'll see today what kinds of genes are involved in this. And those mutations, those 12, you could inherit them from your parents. So you could inherit 8 mutations from your parents, and then you get cancer at 40, because it doesn't take that long to get the other 4. Or you could inherit none, in which case you're really lucky. But it's all about those. And how do you get them as you live well? Your DNA replication is error-prone, so you can sustain mutation. You have 3.2 billion base pairs of DNA, so it's the chance that it's going to be in the mutation in the right gene is kind of low. So some of us never develop cancers in our lifetime. You can sustain DNA damage if you have a job that exposes you to agents that'll do that, carcinogens, smoking, of course, will deliver carcinogens to the lungs, and viruses can do this. And that's what we're going to talk about today. But they all do it the same way, by genetic alterations. So Transformation and oncogenesis are distinct. Transformation is a property of cells in culture, which is absolutely needed for oncogenesis. Oncogenesis, the development of a tumor. And what I will tell you today is a story of how we learned how viruses cause transformation, and then that leads to oncogenesis, and how that informs our understanding of cancer in general. And I say here, no virus can do it all. The virus doesn't actually cause the cancer. The virus transforms the cell, as you will see, and then the cell keeps dividing and making mutations, and then a cancer arises, which is really independent of what the virus does. So we have viruses that are a contributing factor in about 20% of human cancers. And here are the viruses listed with the kind of cancers that they cause. Both RNA and DNA viruses are causing cancers. HTLV1, HIV1, we will talk a little bit more about HIV1, hepatitis C virus. And then among the DNA viruses, 2 herpes viruses, hepatitis B virus, human papilloma virus, and a polyoma virus. The mechanisms we're going to talk about today don't apply to any of these. But what does apply is the general property that if cells divide uncontrollably, eventually they will become a cancer. And that's what happens with many of these viruses. For example, HIV causes the production of huge amounts of cytokines. What do cytokines do? One of the things they do is make cells divide. So those cells that keep dividing develop into tumors. But one very important point here, this is a fish with a tumor on it. It's a walleye fish. In case any of you like to fish, this is what this is. And the tumor is caused by a virus called walleye dermal sarcoma virus. And I used to say in this course for years, no transformation in oncogenesis is not required for the replication of any virus. And in one year, I had to put an asterisk on any, because there is one case where it is. Someone emailed me because they listened to the course, and they work on this virus, and they said, "You're wrong. There's one case where you need to make a tumor, and that's because the virus is in that tumor, and in the fall, the tumor falls off the fish into the water, and that's how the virus spreads to other fish. And the fish is fine in the end. It's probably going to get caught by someone, but the tumor doesn't kill it. But the tumor is needed to spread virus. That's the only case that we know of where transformation and oncogenesis are needed for the replication of the virus, because it needs to find a new host. All right, so the story begins in 1909, right here in New York City. Rockefeller Medical Institute. Peyton Rouse is an MD who wants to do some research, and he's interested in cancer. And a farmer from New Jersey brings him a hen that has a tumor on it. Famous chicken there with a tumor. And so he takes off this tumor. In 1909, we know already filterable viruses, right? We know that you can filter tissues through a certain kind of porcelain filter, and the virus will go through and the bacteria will remain. So he wanted to know what was causing this. Is it a virus or a bacterium? So he ground it up, filtered it through a porcelain filter. He injected the filtrate, and it caused tumors in another chicken. Cancer could be caused by a virus infection. Unfortunately, nobody believed him for many years. He actually stopped working on it because he couldn't convince anyone that he was right. And then later on, evidence began to accumulate that he was right. He returned to it, and many other people returned. And 50 years later, 1966, not too long before he died, he got the Nobel Prize in Physiology and Medicine for this work, showing that a cancer can be caused by a virus. Now, the virus he found is called RSV, Rouse sarcoma virus, named after him, Peyton Rouse. And it gave rise to 2 additional Nobel Prizes, as we will see today. The longest incubation period for a Nobel Prize. Now, this whole story of how it was figured out that this virus, or the mechanism by which this virus causes tumors, is in this very nice book by Siddhartha Mukherjee, who's a oncologist up at the Medical Center of Columbia. I'm sure some of you have read this. He won the Pulitzer Prize for this. In my opinion, it's his best book, The Emperor of All Maladies, A History of Cancer. So he writes, by the 1950s, cancer researchers had split into 3 divisions, and they all argued with each other. One said viruses cause cancer. That was Rouse and others. But no human cancer virus had ever been found. That's always the issue. Epidemiologists said that chemicals caused cancer, but they didn't know how they would do it. And the third said, our own genes could cause cancer. And they didn't know how that would work either. Well, the story I want to tell you today brings all of this together. And many years later, 1951, Howard Temin goes to Caltech to study fruit flies. He got bored with fruit flies, and he decided to go work with Renato Del Becco. In 1951, Del Becco has just developed the plaque assay for animal viruses, and he recognizes the importance of using cells in culture to study viruses. And so Temin wants to do this. He says, I want to use cells to study cancer. I mean, up until this point, people have been studying Rouse sarcoma virus in chickens. And there's only so much you can do in chickens. You can't do any biochemistry or genetics. So Temin said, I am going to reproduce cancer in a petri dish. It's a little bit beyond what he actually wanted to do, but that's the image that you can get. He spent 7 years doing it, and he got it. So what he did, he took chicken cells. You can take a chick embryo and chop it up and make single cells, put it on a plastic dish, and they will make nice monolayers. He added Rouse sarcoma virus to these chicken cells, and he got foci. He got transformed cells. And there are 2 kinds of transformed cells here on the left, kind of long, spindly cells. You can see the background of regular, normal chicken cells there. And then this focus, he called them, of transformed cells. And then you could make round, refractile cells as well. And you could imagine counting these, right? And then you could have a titer of the virus in focus-forming units per milliliter, just like plaque-forming units per mil. And that's exactly what he did. So he said, and these are the words of Mukherjee, Temin, the foci, he said, represented cancer distilled into its essential elemental form, cells growing uncontrollably, unstoppably. And it's true. These are transformed cells, the precursors to cancers, and you can see them piling up there on the right. So this was a huge advance because now people could look in cells and say, what's the mechanism by which the virus is transforming them? Very important. In addition, other people got interested in this, and there were other discoveries. DNA viruses. So polyomaviruses, 1962, it was shown that they could transform cells in culture. And in 1964, SV40. Remember SV40? We talked a lot about someone infected cells and showed that they were transformed again. So the virus kills most of the cells, but a few are left that are transformed and have this property of living forever and all the other properties that we talked about. So the question that we're going to answer today is, how can a virus infection transform a cell? So we have 3 requirements here. First, you have to reduce cytopathic effects or get rid of them, right? Because if you kill all the cells, they can't be transformed, right? Because a transformed cell, by definition, needs to live forever. So somehow you have to reduce CPE. You have to reduce virus replication. That goes together with CPE because the particle production is associated with killing. And the cell has to be immortal. It has to divide forever. And does that ring a bell? It's a kind of persistent infection where the cells, the virus is there, maybe reproducing very slowly, if at all, but the genome is present. And this transformation is basically a special kind of a persistent infection, if you will. All right, our first question is, which of the following is not a property of transformed cells? Increased requirements for growth factors, immortality, loss of anchorage dependence, loss of contact inhibition, colony formation in semi-solid medium. What's not a property of transformed cells? All right, what do we have here? Yes, increased requirements is not a property of transformed cells. We have decreased requirements for growth factors. All of the others are properties of transformed cells: immortality, loss of anchorage dependence, loss of contact and division, colony formation, and semi-solid media, all properties of transformed cells. So here is an overview of what we're going to talk about today, the route to understanding how cells are transformed by virus infection and how that leads to cancer. Very tortuous route to getting there. First, we're going to talk about retroviruses. Rous sarcoma virus, which he first found in 1909. I told you Temin figured out how to study these viruses in cells in culture in vitro. Then on the right, DNA viruses that were found to transform cells, in vitro studies in cell culture. And then finally, studies of the biology of cancer cells and how that relates to this. They all converge in the 60s and 70s. Our present understanding of the control of cell growth comes from all of this. Without viruses, we would not understand how cell growth is controlled and how it is aberrant in cancer. So let's start with, or go back to the retroviruses, because Rous sarcoma virus was called initially an RNA tumor virus. And remember, reverse transcriptase was discovered in the early 70s in that virus, so they were then renamed retroviruses. So the virus that Rous worked with is derived from a chicken retrovirus called avian leukosis retroviruses, or ALVs. These are in all chicken flocks in the world. Every chicken pretty much is infected with this virus. It was discovered in 1908, the year before Rous discovered RSV, to cause leukemia in chickens. But at that time, people didn't think leukemia was a cancer because it wasn't solid. It didn't make a big tumor. So it's aberrant white blood cells. It's not a cancer. We realized that later, of course. But that's why we talk about Rous discovering cancer viruses. So most chickens are infected with this virus within a few months of their hatching. About 3% of birds over 14 weeks of age develop leukosis, which is another word for leukemia. It happens randomly. And the rest of the birds, 97% of them, they have a transient viremia. They become immune. They don't get leukemia. So a very small proportion of the infected chickens develop leukemia. And by the way, this is all chickens. Even the chickens you're eating, they have these viruses in them. Can't stop it, but they're fine. They're not going to infect you. As these birds age, they develop other cancers. They develop connective tissue tumors or sarcomas. These are solid tumors of connective tissue. So these infected birds, as they age, initially they develop leukemias, and then the others develop solid tumors. And if you take the virus from those solid tumors, as Rous did, and put it back into a chicken, you get a solid tumor. You don't get a leukemia, even though, as I will show you, these are derived from avian leukosis virus. And one of the viruses that Rous isolated, Rous sarcoma virus, is the causative agent of a solid tumor. So in aged chickens, they're developing these solid tumors. Most of these viruses, and many people isolated different viruses over the years. Everyone had their own chicken with a tumor because you can get them from any chicken farm anywhere, and they isolated different viruses from them. Most of them were defective, actually. They couldn't reproduce on their own. What does that mean? There's a deletion in the virus genome, so they're missing a part of a gene, and they need a wild-type helper virus, the same virus, to allow it to reproduce. But Rous was lucky. His virus was not defective. Oh my gosh, everybody after him isolated a defective virus causing sarcomas of chickens, but he didn't. So, you need luck sometimes. Although, as Pasteur said, luck favors the prepared mind. Maybe his mind was prepared. Who knows? Anyway, how does Rous cause solid tumors but not avian leukosis virus? Well, people were interested in that. People started comparing the genomes of the two viruses, right? Rous and ALV, Rous solid tumor, ALV leukemia. And these two individuals found the difference. And they got the Nobel Prize for that in 1989, Michael Bishop and Harold Varmus. His first Nobel Prize, so the second Nobel Prize for Rous sarcoma virus. they found that a piece of the ALV genome is replaced with a piece of host DNA, which was called an oncogene because it lets the virus cause tumors. So the oncogene they called V-SARC, SARC for the kind of tumor, sarcoma, that it causes. They did that in 1976. They got the Nobel Prize in 1989. Harold Varmus, he's here at Sloan Kettering, still working on cancers. He's working on sarcomas in humans, actually, and he was the head of NIH for 10 years before that. I've had them both on my podcast separately, so those are the links to that. Very interesting individuals. Okay, so what happened to ALV? Somehow a piece of the host DNA got into the virus, and now that virus is Rous sarcoma virus, and it causes a solid tumor in chicken. So this is the major insight from all of this work, right? ALV-infected birds, if you let them age, right? If you don't kill them at a year for meat, if you let them age, they get tumors. The tumors all have retroviruses derived from ALV, but if you pick different viruses from different chicken tumors, you can see they're all picking up a slightly different gene. They're picking up the same gene, putting it in a different place in the genome, and they're all slightly different. And as I said, Rous, his isolate was not defective. And I'll show you a picture of this in a moment so that it'll clarify what I'm talking about. So each of these viruses that were studied, in fact, had different host DNA in them. I just misspoke. Not the same host DNA, not VSARC. They had a different piece of DNA from the chicken, and each of those genes was called an oncogene. So apparently within the chicken genome, and presumably of every other mammal and bird, there are sequences that can cause cancers if they're put in a retrovirus and reintroduced into cells. So these are called oncogenes, and this was a gold mine for studying how viruses transform cells and how tumors arise, in fact. So here's a map of all of these, what we call transducing retroviruses. Transducing means the retrovirus infects a cell, picks up a piece of cellular DNA, and then it brings it to another cell when it infects it. So on the left are the avian transducing retroviruses. So we've been talking about chickens, right? So here's Rous sarcoma virus. On the top here is avian leukosis virus. So that's the virus that infects every chicken in the world. And there you said it's a typical retrovirus. It has a gag, a pole, and an envelope gene. And look at Rous sarcoma virus. It's picked up SARC, which is the cellular gene that causes it to transform cells. And you may be thinking, why would a cell gene do this? And we'll get to that because it's an interesting question. And below Rous are all the other transforming retroviruses that other investigators identified after Rous had done his. And you can see they're associated with different kinds of tumors, including sarcomas. And they have different oncogenes picked up from the host cell, MIB, MIL, YES, ERB, etc., which are named basically after the kinds of tumors that are produced. And we can do the same thing in mammals. You start with a murine leukemia virus, which infects many mice globally in the wild, gag, pole, and envelope. And you can see transforming derivatives of it have different oncogenes from the mouse integrated into the retrovirus. And I want to make sure you understand here that most of these are defective, right? So SARC was stuck on after gag, pole, and envelope in Rous. But you can see MIB has disrupted the envelope gene. So now this virus cannot make a glycoprotein, so it needs another virus to infect the same cell in order to be packaged. So many of these are defective, and that's what I mean by that. So defective versus non-defective, we need a helper virus. So here is avian leukosis virus. That would be the helper virus for avian myeloblastosis virus BA1, for example. So here we have Rous, which is not defective because SARC was stuck on at the end of envelope just by chance, but MIB has integrated into envelope. So this virus, AMVBA1, will only reproduce in cells co-infected with ALV. But in a chicken, that can happen because they're infected with both ALV and the BA1 virus. So that's what I mean by defective retroviruses. So how does this happen? How does a retrovirus pick up a piece of host DNA? So here are two mechanisms. I'll just show you one to give you a flavor. So remember, retroviral RNA in the particle is reverse transcribed to DNA, which then integrates into host cell DNA as a provirus. So after step one here, we have proviral DNA. There's host DNA on either side. And let's say this provirus integrates next to an oncogene, which is one of these genes that we have defined, if picked up by a retrovirus, will cause transformation of cells. So here's an oncogene. And a certain fraction of the time. Remember, transcription occurs from the left-hand LTR, and so you get retroviral RNA. Sometimes the transcription will keep going past the right-hand LTR. Maybe sometimes the right-hand LTR is deleted by recombination. So now you have an mRNA that contains the viral proteins, but also the oncogene sequence in there. And that's going to get packaged into the particle. And eventually, when that virus infects a new cell, it's going to deliver the oncogene to the cell under the control of the retroviral LTR, which means high level of transcription now, no regulation that you would normally see with this gene in the cell, and that's how these viruses transform cells, because the oncogene perturbs cell growth, as you will see in a moment. So the cell, from studying all these different retroviruses, transducing retroviruses, we have identified over 60 genes within the cell, which we call oncogenes. It's not a good name. They're not there to cause cancer, right? But that's how we found them, because, Rous sarcoma virus caused cancer, so we called the gene it picked up an oncogene. But in the cell, these are genes involving regulation of cell growth. And cell growth is highly regulated. Here's a diagram of the mitosis, the cell division cycle, right? You have mitosis for a very short period out of the 24 hours, then a growth phase, a DNA synthesis, or S, and then a second growth phase, and then the cells divide. Cell growth is highly regulated in cells because you don't need every cell to be able to grow all the time. And these genes regulate cell growth in ways I'm going to show you in a moment. And so the nomenclature for these genes, the cellular genes, they have a C in front of them, like C-SARC, C-MYC, C-MOS, C-RAS, etc. And then certain retroviruses from tumors have these genes integrated into them, and we call them V-SARC, V-MYC, etc. That's the nomenclature. But as I said, I think oncogene is a bad name because they're not there to cause cancers in you, right? They have other functions. So here is a series of oncogenes identified in a variety of viruses of primates, cats, chickens, mice, more chickens, that have picked up cellular genes, oncogenes, and here on the right is where those genes are active in the cell growth regulatory pathway. It's just amazing. So cell growth happens when we have a growth factor that binds to a growth factor receptor on the cell surface. So you're adding serum to your cells to make them grow. Serum has growth factors in it. They bind receptors. That binding causes a cascade of phosphorylation events that ultimately lead to the turning on of transcription in the cell nucleus of all the genes you need for the cell to divide. There are many. So that whole pathway, every component of that pathway, the growth factor receptor, the tyrosine kinases, the cellular proteins, the signaling proteins, the transcriptional regulator, the genes for each one of these has been picked up into a different retrovirus. And so, for example, let's say one of the transcriptional regulators, if you are producing it in a retrovirus and overproducing it, the cell is going to be dividing because you're turning on all the genes that you need for division. So you see, by inserting a gene for any one of these intermediates into a retrovirus, you turn the retrovirus capable of making the cell divide. Now remember, that's the original phenotype we saw in cultured cells, uncontrolled division transformation. And that's caused by inserting either any of these proteins, genes encoding these proteins, into a cell via a retrovirus. So it's really remarkable. And they figured out the whole pathway just by pulling out retroviruses of different species. So again, here is the cell cycle. There is a go signal at the top. which is encoded in these proteins that I've just shown you whose genes have been picked up by transforming retroviruses. They are mitogenic signals because remember, when we add growth factors to cells, they bind receptors and get the cell eventually to divide, and that's the go signal up here. And all of the components of that go signal were identified by studying these transforming retroviruses. We call them dominant oncogenes. You just need to put one copy in a cell, and it will start to divide forever. And remember, they start dividing. They become transformed. Every division, they sustain mutations. And if you get enough mutations, that cell, if it's in a host, could start to become a cancer cell. Now, it turns out that the transformation by retroviruses is a little more nuanced than what I've told you. There are actually three ways that retroviruses transform cells. We have rapid tumor formation, which like about 2 weeks. You inject Rouse sarcoma virus into a chicken, and you have a tumor formed in 2 weeks. And that's because the virus genome contains this oncogene in it, coding sequence, and as soon as the virus infects the cell, the protein is produced, and the protein drives the replication of the cell. Rapid tumor formation. Intermediate kinetics. Some retroviruses take months to make a tumor, and an example is avian leukosis virus, right, which takes a few months in the chicken, and that's because the virus does not contain an oncogene like Rouse. There's no oncogene. Rather, the virus integrates next to an oncogene and turns it on. And that just takes time. And finally, there are retroviruses that take years to cause tumors. And an example is HTLV, human T lymphotropic virus, which can cause cancers in humans. No dominant oncogene, does not insert next to an oncogene, but rather it encodes a regulatory protein, a transcriptional regulatory protein that's needed to turn on viral transcription. And that protein happens to also turn on cellular genes that are involved in, cell division. So that takes a lot longer. So the three different kinds, and here are just diagrams to show that. Rapidly transforming cells, the virus genome carries a oncogene. Intermediate transformation, where the retroviral genome, the provirus, integrates next to an oncogene, and the promoter in the right-hand LTR turns on transcription of the oncogene. And finally, the transactivating, there's no oncogene in the genome. It doesn't integrate next to an oncogene, but one of the viral proteins here, it's called X, which is needed to transcriptionally activate the LTR, will actually hit a cellular gene, for example, like IL-2, which is a cytokine that will cause cell division in T cells. And that causes them to divide forever, and eventually you get transformation of the T cells, and they form tumors. So all of these viruses transform cells as a mistake. As I said, there's no need for the viruses to do that in the context of a reproduction. They don't need to do it to make infectious particles, but it's a mistake because they have to integrate. The genome has to integrate, and so by chance, you're going to go next to an oncogene, or you're going to pick up an oncogene, and so forth. So there's no requirement for transformation except for walleye dermal sarcoma virus. Our next question is, which of the following allows Rouse sarcoma virus to transform cells? Presence of the envelope gene, presence of the Paul gene, presence of a Sark gene, presence of LTRs, or none of the above? Which allows it to transform cells? All right, let's have a look here. It's the presence of a Sark gene. Most of you got that. That's the transforming gene that makes the virus transform cells. So the envelope, the Paul, the LTRs, those are not causing it to transform cells. The introduction of the Sark gene is what does it. I think you see now how studying these transducing retroviruses illuminated the control of cell growth. Same thing happened with DNA viruses starting in the 1920s. This begins with Richard Shope. His son was Robert, who was also a virologist. Richard Shope finds a DNA virus that causes warts or papilloma. So a papilloma is another name for a wart in cottontail rabbits. So here are two rabbits in the wild that have very scary looking warts on them caused by this papillomavirus. And yes, human papillomavirus causes warts in humans as well. These rabbits are fine. The warts fall off eventually and they're okay. But if you've heard of the jackalope, which is a mythical creature, a jackrabbit with antlers, this is what a jackalope is. People see these, they think they have antlers, and they think it's a hybrid between a jackrabbit and an antelope. But it's not. They just have papillomavirus-driven warts. Also, Ludwig Gross, working separately, found that certain polyomaviruses caused tumors in mice. These viruses, the natural host is the mouse. They don't cause cancers in mice. But if you infect hamsters or rats or rabbits, the wrong host, they make a lot of different kinds of tumors. That's why they were called polyoma, many different tumors. And then finally, in 1962, Maurice Hilleman and Bernice Eddy showed that SV40, which we've talked about a lot, induced tumors in hamsters. And this virus is a virus of non-human primates, does not cause tumors in monkeys, does not transform monkey cells in culture, but if you infect the wrong host, hamsters, you get tumors. And this is going to be an important part of understanding what's going on here. By the way, the earliest polio vaccines were made by taking kidneys out of non-human primates, making cells out of them in culture, and growing poliovirus in them. We didn't know that these cells had SV40 in them. We didn't know SV40 existed in 1950s. So many millions of Americans were given polio vaccine with SV40. I probably got it because I had my first polio vaccine in 1955 until we figured out that it was there. It doesn't look like it caused any problems in people, although if you search, you'll find lots of lawsuits, of course, by various people who think their tumors were caused by SV40. So here's a summary of what I've told you. We have different species on the left, and we're infecting them with either SV40 or a mouse polyomavirus. So SV40, the host species is the monkey, and those cells are completely permissive to infection. And when you infect monkey cells with SV40, it kills them. It kills all of the cells, doesn't cause any transformation. If you infect monkey cells with a mouse polyomavirus, the cells are non-permissive. The virus doesn't even replicate. That's what permissivity means, right? So you never have any tumors. Tumors on this chart, by the way, are shown by the red asterisks. Now, if you take the mouse, SV40 is non-permissive in mice, so you don't get anything. Mouse polyoma is completely permissive in mouse cells, so the virus reproduces and kills the cell, so no tumors. Now, if you infect hamsters with SV40 or mouse polyoma, or rats with SV40 or mouse polyoma, in both cases, the infection is semi-permissive, and you get tumors. And semi-permissive means you get up to DNA replication, and that's it. So you get early gene expression, but no late gene expression. And early gene expression is apparently enough to transform the cells and get tumors. That's what semi-permissive means. And this kind of transformation of cells in culture, where we infect with, say, a polyomavirus, and we look for foci, it's pretty rare, 1 in 100,000. Why? So let's examine why it's rare, and how does that relate to rare tumor formation in animals. First, adenovirus, another DNA virus found to cause tumors. So far we've found papillomas and polyomas, now adenoviruses. These are human viruses, many serotypes. They do not cause tumors in us, but they will cause tumors in hamsters. And they're rare events. And again, the wrong host, where the host is semi-permissive for infection. So what's in common among these three, papilloma, SV40, and adenovirus? In transformed cells and tumors, people found T antigens. They call them tumor antigens. That's where the T comes from, that we talked about a long time ago with SV40. The T stands for tumor because the proteins were first discovered in transformed cells and tumors. For SV40, it's large T. There's also a small t we haven't talked about. For polyomas, there's a large, middle, and small. For papillomas, T are encoded by the E6 and E7 genes. And for adenoviruses, they're the E1A and the E1B proteins. So these proteins are all found in tumors and transformed cells, but they're all different proteins. They're not the same protein encoded in different genomes of different viruses. Now, as you know, T antigens are essential. We talked about the requirement for DNA synthesis, the requirement to activate transcription, and the genes encoding these proteins, T antigens, E1AB, E6 and E7, they are the only genes that are always retained in either transformed cells or tumors. So if you look at a lot of cells transformed, say, by any of these viruses, the only thing in common are the genes encoding these T antigens. And so, in fact, if you take the gene for T antigen, for E6, E7, for E1AB on a plasmid and put it in cells, you can transform the cells. So, if you want to take your cheek cells and immortalize them, just put in T antigen. You could put telomerase in too, or T antigen. T antigen will make them divide forever. And this is something you know already, but I will remind you of this. Then people did additional work, of course, and they made 3 discoveries that made the connection between these viruses, the transformation, and the cell cycle. First, a 53 kilodalton protein was discovered to bind T antigen. This is the now famous P53. That is the gene for which is mutated in nearly every human cancer. Secondly, and you've heard this one before, but I don't think you're going to remember, transcription of the E2 genes, the early region 2 of adenovirus, requires a cellular transcription factor that we've called E2F. Remember, that's needed, and that is freed up by binding to retinoblastoma. So E2F binds RB. So T antigen binds P53, E2F is needed for adenovirus transcription, and E2F binds RB. All these 3 proteins are controllers of the normal cell cycle. So here's your cell cycle again, just to remind you, you have a 24-hour cycle during which a very small part, the cells undergo actual mitosis. But there's a period, after mitosis, there's a period of cell growth. So you've now divided the cells into 2. You have to let them grow. Then you have to replicate the DNA. You have to duplicate it so each cell will get an equal amount. And then the cells prepare for division. They then divide. So when you stimulate a cell to divide up here at the top by those retroviral oncogenes, they go through the cycle, they replicate their DNA, and they prepare for cell division. And remember that the regulation at the top here is controlled by growth factors, growth factor receptors, all the genes that were picked up by transforming oncogenes. That's what told us how this control works. And essentially, if there are no growth factors in the medium, the cell's not going to divide. The light turns red. You need a growth factor to bind the receptor and push the cell through, because without that, the medium is not rich enough to sustain cell division. There's also another restriction point down here, sort of in the middle of G1 here, which will say, okay, you pushed me from the control point at the top here, but are conditions actually right for going through to the cell division cycle? The protein that regulates this checkpoint, so these we call checkpoints, there's one up at the top and there's one at the bottom here, is RB, retinoblastoma protein, which we've encountered before. This was found initially in young children who developed tumors of the retina, in the retinoblast cells specifically. They are gone, those cells are gone by age 5, so all these tumors you see in very young kids, and they were found to have a deletion of the gene encoding the RB protein. And so RB regulates that division. If you lack RB, your cells just keep dividing forever, and you develop retinal tumors if it's in fact those cells in which the RB gene is deleted. So RB is an important checkpoint. Now, in the cell, there are mechanisms to get RB deactivated, and that's by phosphorylation, as you'll see right here. So here on the left is the mitogenic pathway. You have a receptor protein, tyrosine kinase, which binds a growth factor receptor, for example. The growth factor binds, causes phosphorylation of the receptor, and then a cascade of events leading to the turning on of the genes that are involved in cell division. That's the G1S transition. The cells make DNA and they divide. So this is a whole signaling pathway stimulated by growth factor receptors. And a key part of it is the RB protein. When RB is phosphorylated, it's bumped off another protein, the E2F protein, and that turns on the transcription of all the genes that are needed for cell division. So here on the right, here's RB bound to E2F. Just to remind you, RB recruits histone deacetylases to the promoter of all these genes, and that represses transcription by tightening up the histones. If you phosphorylate RB, it comes off of E2F, and now these proteins can bind the promoters of genes that are needed for basically DNA synthesis and cell division. And there are many, many such genes. But RB is a key regulator. If the conditions in the cell aren't right, and that's signaled from the growth factor receptor as shown here, RB will remain unphosphorylated and the cells will not divide. So what does it have to do with viruses? Well, remember, viruses, especially DNA viruses, have to have dividing cells in order to replicate their DNA, especially the viruses with small genomes. They don't encode a complete DNA synthesis machinery. So when they get into a quiescent cell, they have to make it divide. And how do they do that? Well, it's the T antigens. The T antigens bind RB and bump it off E2F, and now all the genes needed for cell division can be activated. T antigens kick quiescent cells into S phase. They relieve this checkpoint here in G1 and allow the cell to keep dividing. That's so they can make viral DNA. And so here's a picture of that. You have seen it before. Here is E1A, large T, E7. These are the tumor, the T antigens that are common to transform cells, that are common to tumors transformed by these viruses. Those proteins bind RB, and that frees up E2F so that it can turn on DNA synthesis and other proteins, transcription of proteins involved in cell division. So essentially, the T antigens have a similar function as phosphorylation of RB. Phosphorylation of RB is done by the cell. The binding of RB is done by these viral proteins. So the viral proteins bind RB, and now the cell cycle can proceed. Right? That's the normal function of these T antigens in virus replication. One more explanation before we sum all of this up. The entry into S, the DNA synthesis part of the cell cycle, has one more control. So we've talked about RB control, and that is P53. P53, the protein found to bind these T antigens. P53 will detect unscheduled DNA synthesis. So what does that mean, unscheduled? You don't have an appointment? It means it's not cellular. It's not happening by the cell. A virus has come in and has attempted to turn on DNA synthesis. P53's function is to detect that. And there are a whole series of detector proteins that detect, for example, double-stranded DNA breaks, which are common in viral DNA synthesis. And when P53 detects that, it will initiate an apoptotic cycle in the cell cycle arrest, apoptosis, and the cell dies. So you kill the cell to spare the organism. P53 needs to be countered, otherwise these viruses are not going to have their genomes replicated. And so that's another function of these T antigens. They bind and do various things to P53. These are different ways that viruses counter P53. And I just want you to know that, the papillomavirus protein binds P53 and causes it to be degraded. The SV40 large T binds P53, so does E1B of adenovirus, and sequesters it so it can't have its normal activity. So the T antigens sequester RB and get the cell cycle going. P53 tries to intervene. The T antigens bind that as well. So now the cell is able to undergo DNA synthesis and go through the cell cycle. All right, so those are two important controls that are both alleviated by these amazing viral proteins. Why? Because they need to be able to reproduce their genomes. Last question. T antigens are encoded by viral genes that are essential for replication, present in tumors and transformed cells, encoded by viral genes that have been incorporated into the cell genome, antagonists of cell cycle checkpoint proteins, all of the above. What are T antigens? All right, what did we do? So all of the above is correct. These are all properties of T antigens. They're encoded by viral genes. They're essential for replication. They are present in tumors and transformed cells. They're encoded by viral genes that have been incorporated into the genome, and they're antagonists of checkpoint proteins in the cell. All right, so two more things we have to explain. Why are only T antigen genes found in cells transformed by these viruses? Everything else is gone. And why is transformation so rare? So the last question is, transformation is rare because we have two low probability events that have to happen. First, the lethal late genes can't be expressed. Late genes encode the capsid proteins, and they would kill cells. So when a virus transforms a cell, there's typically been a rare deletion of the late genes. It's just spontaneous deletion that happens randomly, and that allows the cell not to be killed. So they're semi-permissive now. Or you infect semi-permissive cells to begin with, where you never get into the late phase. So a primate virus in a mouse cell, the mouse cell is semi-permissive. It will get in and go up to DNA synthesis and stop. And so you will not have the cell die, and that's how it can be transformed. So that's first. You have to have the suppression of the lethal late genes of the virus. Secondly, the T antigen has to be in every cell. It has to be on in every cell, which means that the DNA has to integrate, and that's a rare event also. So a virus infects a cell, the DNA is going in the nucleus. A certain percentage of the time it will randomly integrate into host DNA, and if it does, and the host is not killed, then that cell will keep dividing forever. And so this is all an accident because people were using the wrong hosts. These are not these events, the causing of tumors, transformation of cells and causing tumors, don't happen in the normal hosts for these viruses because the viruses kill cells. So we discovered them by putting the virus in the wrong host. Why did we do that? Well, someone once took a polyomavirus and infected a different host just to see what would happen. And they got tumors. And that led them down this path. So it's a nice way of saying, if you have an idea, just try it. Who knows what's going to happen? So these are rare events that I've been telling you about in these cells infected with DNA viruses. But we now understand that in the natural host, you can get transformation. So human papillomaviruses cause tumors in us. We are the natural host for human papillomavirus. So it doesn't always have to be the wrong host. That was a good experimental trick to figure it out. So I say that transformation is an epiphenomena of unique reproductive cycles of these DNA viruses. The DNA viruses have to make the cells divide, so they make DNA. The T antigen turns on division. They inactivate RB and they inactivate P53, and this allows the viruses to reproduce. But if somehow the cells survive and the DNA encoding T antigen gets integrated, the cells divide forever. They're transformed, and as they divide, they sustain mutations. If they get that magic 12 mutations, boom, they are a tumor. So the virus is not participating in that last part, if you will. So here's your question about HeLa cells. So this is a wonderful book you should read, The Immortal Life of Henrietta Lacks. So Henrietta Lacks had a cervical tumor in the early 50s. Her surgeon took out a piece of it and put it in culture, and it turned out to grow forever. The cells were transformed. They became known as the HeLa cells. And many years later, the genome of HeLa cells was sequenced, and lo and behold, there's human papillomavirus type 18 integrated into her chromosome 18. But only 2 genes were present from the virus. E6, which binds RB, and E7, which degrades P53. So the 2 that you need to get DNA synthesis going for the virus, this piece of DNA got integrated by accident, and it caused her to have a tumor. She eventually died as well. This is very rare in infected people. Many people have infections with human papillomaviruses. Only certain of the types cause cancers, but they're also quite rare as well. So you notice that the name of the cell, HeLa cells, comes from the first 2 letters of the first and the last name. I have a son who is an amateur disc jockey here in the city, and he made his name Ira, because his name is Aiden Racaniello, because many years ago I went to his high school and taught them about HeLa cells, and so he decided to make his DJ name Ira. But what's even more cool, he's playing this Saturday here in New York City at the Feast of the Jackalope. So 2 things from this lecture, right? There's a jackalope, this mythical, antelope jackrabbit, which is nothing more than a papillomavirus causing papilloma. So here we have a reference to, in 2 different ways, to papillomaviruses. So I had to show it to you. I might go, but it's like till 5 a.m. I can't take that. All right. So to summarize, we have looked at RNA tumor viruses and DNA tumor viruses to sort out the cell cycle. And in both cases, the transformation is a side effect of a unique reproduction cycle. Retroviruses have to integrate into the cell, and that can cause an accident now and then. You can integrate next to an oncogene. You can pick up an oncogene, as we've seen today. And those oncogenes turn on the cell cycle at this very top checkpoint here. DNA viruses, of course, and those retroviral oncogenes are called dominant oncogenes. You just need 1 copy of the DNA to have the effect. DNA viruses have to get cells dividing. They do so by interfering with RB and P53. And if by accident the virus does not kill the cell and those genes, the genes encoding those proteins, are integrated, the cell keeps dividing and it eventually becomes a tumor. And we call these recessive oncogenes, the P53 and the RB. RNA tumor viruses integrate, DNA tumor viruses turn on the cell cycle. These both lead to these aberrant transformations. In the process of studying and understanding them, I think it's really cool that we understood the cell cycle. By the way, the tumor suppressor genes encoded by DNA tumor viruses, of course, hit that second checkpoint, the RB checkpoint, down at the bottom of the cell cycle. And the amazing thing is this started out with a guy here in New York trying to understand a tumor of a chicken. So next time we will start talking about trying to interfere with virus infections, and we're going to start with a discussion of viral vaccines.